I am Toriumi, Managing Officer of the Department of R&D of Bona Corporation. I'd like to thank Governor Ogawa of Fukuoka Prefecture and distinguished members of One Health International Forum for giving me this opportunity to speak to you. My talk today will cover the topics on SARS-CoV-2 that caused COVID-19. And these are the contents of my talk, as you can see on the slide. That includes current strategy for development of anti-SARS-CoV-2 therapeutics, research of siRNA therapeutics for viral disease including SARS-CoV-2, current status of development of siRNA therapeutics for SARS-CoV-2 in BONAC. And finally, I will talk about potentials of siRNA therapeutics for emerging viral diseases. Please allow me to briefly introduce our company. Our company was established in 2010. We are a bioventure specializing in nucleic acid chemistry, headquartered in Kurume, Fukuoka. As we will discuss this in more detail later, the most advanced product in our development pipeline is TRK-250, which is nucleic acid inhalation medicine for idiopathic pulmonary fibrosis targeting EGF-beta-1. As of November 2010, when this presentation was prepared, globally speaking, we see an increase in daily new cases and the daily death is around 5,000, and there's no prospect of this being coming to an end. Under these circumstances, researchers and scientists all over the world are working on COVID-19 coronavirus therapy. This slide shows drug discovery-related molecules at different phases, including entry, replication, and release of virus. Entry of virus, genomic transcription, protein translation, and virion assembly are included. There are various candidates that could selectively inhibit these steps. However, remdesivir, shown in red, is the only drug approved so far among these promising medicines. And this is the schematic of the steps shown earlier, including viral attachment to cell entry, replication, and release. Remdesivir, which is already approved, and Abigan, soon to be approved, inhibit RNA-dependent RNA polymerase. And they exhibit antiviral effect by inhibiting viral RNA replication. So viral RNA replication inhibition can be considered as a promising therapeutic approach. The slide shows the reported side effects of drug-drug interaction of remdesivir, which is already approved, and other new drug candidates. As stated in COVID-19 guideline, hepatotoxicity should carefully be monitored when using remdesivir. For favipiravir, Abigan, in addition to hepatic and gastrointestinal adverse effects, teratogenicity was confirmed in non-clinical study, so pregnant women should not be treated with this drug. So there will be a continued need for SARS-CoV-2 therapeutics free of adverse events. Under these circumstances, RNA interference, in particular small interfering RNA or sRNA, is getting attention as a new therapeutic platform. The slide shows number of deaths of SARS-CoV in 2002 and WHO data on SARS-CoV-2 as of May 2020. A number of PubMed publications on sRNA and cancer and sRNA and SARS. The relative numbers of published papers in the two fields started to increase with the occurrence of SARS-CoV, and it shows the increased interest in applying RNAi technology to SARS-CoV infections. Next, I'd like to talk about the gene silencing mechanism of siRNA. siRNA is a double-stranded RNA with 21 to 23 base pairs. siRNA, through messenger RNA destruction, inhibits gene expression in a sequence-specific manner. This is the mechanism that evolved as a part of bioprotection against viral infection. Long-chain single-stranded RNA in the cell and extracellular double-stranded RNA are cleaved by DISA and converted to low molecule of single-stranded RNA with 21 to 23 base pairs. By helicase enzyme, double-stranded RNA is dissociated to single-stranded RNA and forms complex with algonode protein, which shows endonucleic activity toward target messenger RNA, and cleaves target messenger RNA and blocks protein translation. RNA cleavage with siRNA as a means to knock down gene was first used in basic research for biology and therapeutics. However, more recently, there has been active therapeutic development. This shows the clinical activities of RNAi therapeutics. Onpattro by Alinam in the United States, which targets transthyretin familial amyloid polyneuropathy, and Givlaari or givosiran for acute hepatic porphyria have been approved. There are many late-phase clinical studies on siRNA targeting rare diseases are underway. Though it's not shown here for it being in early stage of development, siRNA for hepatitis B, a viral disease, is also underway. Now, let's look at siRNA R&D on coronavirus. Research is underway for gene silencing against various coronaviruses as an application of siRNA technology. The slide showed some examples. The target virus of siRNA includes SARS as well as MERS, which was first reported in 2012 from Middle East, which has not yet been ended. In addition to human coronaviruses, siRNA is studied for porcine Delta coronavirus and porcine epidemic diarrhea virus. For SARS-CoV-2, at the time when the talk was prepared, there were several papers on sRNA sequence design using computers, but there was no paper reporting antiviral effect of siRNA on SARS-CoV-2 found. This is an example of sRNA effect on coronavirus. This is the preventive and therapeutic effect of siRNA on experimental SARS-infected monkeys. Authors designed siRNA against spike coding and ORF1b and sp12 region. Virus with the titer of 10 to the power 5 TCID50 was intranasally introduced to monkeys. Two types of cocktails were studied for such regimens as prophylactic treatment PL, treatment with carrier CD, post-exposure treatment PT. Fever and pulmonary pathohistology were studied. Regardless of dosing timing and with or without carrier, there was a statistically significant improvement in all items compared to viral infection control IC and non-specific siRNA control or NS. This slide shows mechanism of anti-coronavirus by siRNA. As shown before, siRNA degrades messenger RNA. Through such mechanism, degrades viral RNA and inhibits the translation of a protein necessary for viral replication from messenger RNA. The target messenger RNA are genomic RNA and positive RNA, which are found on upstream and downstream of the target of remdesivir approved for therapy. So it is expected to be clinically useful. Let me explain SARS-CoV-2 siRNA inhalation drug development at our company. In China, in Wuhan, the unusual pneumonia case was reported on the 31st of December, and WHO named this coronavirus as SARS-CoV-2 on the 11th of February. And on the 13th of January, genome sequence was registered in GenBank. We initiated our project in February and began designing and screening siRNA based on this sequence information. As you can see, first in vitro screening used the plasmid with reporter and target gene. Second and third in vitro screening with knockdown and PFU assay used SARS-CoV-2. In parallel with this screening, in vivo stability was confirmed with human bronchoalveolar fluid and optimized it by chemical modification. In the second screening, nucleic acid was introduced into fresh cell using gene transfection agent, and 24 hours later the cell was infected with the virus. Cells were collected 24 and 48 hours after inoculation, and viral RNA was quantified using RT-PCR. Cultured supernatant collected 24 and 48 hours after inoculation was added to fresh cell, evaluated the impact of nucleic acid on viral titer using PFA assay. And the data on the right panel shows siRNA study on influenza virus we conducted before as an example. The screening was conducted in collaboration with Fukuoka Institute of Health and Environmental Sciences using their BSL-3 facility. siRNA was confirmed on the knockdown activity against virus, and it is incorporated into pshRNA type of nucleic acid of which we have an IP protection. Our RNA interference platform is single-stranded, unlike traditional double-stranded RNA, and it self-anneals into double-stranded structure. So the synthesis is more efficient compared to traditional siRNA, where sensor and antisensor are separately synthesized for annealing. With the proline linker at the hairpin, nucleic acid forms pleated structure, and improved stability against RNA enzyme is expected. This slide is the typical knockdown test results of viral gene using Vero E6 with SARS-CoV-2 isolated at Kanagawa Public Health Institute. Test nucleic acid was introduced into Vero E6 with reagent. After 24 hours, virus is inoculated and cultured for 24 or 48 hours. At the end of the culturing, viral load in the collected cells was quantified with RT-PCR. This shows the result of RT-PCR 48 hours after inoculation. These three nucleic acids inhibit viral gene expression in the concentration-dependent manner. IC50 is less than 1 nanomolar. Maximum dose reaction is over 99.7%, and it shows strong inhibition activity against viral expression. As shown by previous slide, our candidate nucleic acids have been showing strong inhibition activity against viral expression in vitro, and with chemical modification, its stability in the biofluid has been improved. Since the target sequence of candidate nucleic acid is maintained in the 99.9% of mutants reported so far, so they are expected to be effective for many mutant strains. It is confirmed that in some of the candidates, the sequence is also kept in SARS virus. Currently, we carry out non-clinical studies for these candidates so that we can start clinical trials as early as possible. Next, I'd like to talk about the clinical development direction of our products at this point. Our target population includes mild patients and moderate patients in class 1 who have preserved respiratory function. However, based on our consultation with the experts, we may expand our target population to moderate class 2 patients. As for the route of administration, as I mentioned earlier, inhalation is what we plan. Because with this, we can deliver the drug directly to the pulmonary lesion, which is the major area for aggravation of the drug. And for mild patients, we plan to use nebulizer. For class 1 moderate patients with the respiratory help, we consider the use of inline nebulizer. This inhalation therapeutics is TRK-250, and it is a TGF-beta-1 targeting nucleic acid medicine. Our development partner, Torii, is conducting a phase 1 study involving IPF patients in the United States. We hope to make most use of this experience for efficient development. One of the challenges of nucleic acid medicine development is the cellular uptake. Cellular uptake of our nucleic acid TRK-250 when administered to the airway of human TGF-beta transgenic mouse, which is IPF model, has been confirmed, and in vitro, without transfection agents, uptake into various pulmonary cells has been confirmed. As a mechanism of cellular uptake, we obtained data suggesting the involvement of macrocytosis and caveolin-dependent endocytosis. From these, we think it highly possible that our nucleic acid candidates can be taken up in the COVID-19 lungs. So far, I talked about our nucleic acids under development. Since this is an academic forum on One Health, I'd like to discuss the importance of One Health from the perspective of coronavirus drug discovery. The left panel shows the coronavirus, and on the right are types of coronavirus infectious to certain animals. There are 4 genera: alpha, beta, gamma, and delta. Alpha and beta are from bats, gamma and delta are from birds. Human infecting virus belongs to alpha and beta. Genus Beta contains 4 lineages from A to D. SARS-CoV-2 and SARS belong to lineage B, and MERS belong to lineage C. Our nucleic acid under development could be effective for SARS-CoV-2 and also effective for SARS in the same lineage B because of the designed sequence that I referred to earlier. But because of low gene sequence homology, antiviral effect against MERS in lineage C of the same genus Beta cannot be expected. However, we are currently working on the wide-spectrum siRNA effective for beta as well as for alpha coronavirus based on novel and traditional RNA design principle. We hope to utilize alpha and beta coronavirus that infect humans and other animals to validate its antiviral effect. Since nucleic acid manufacturing cost is still expensive as they are low molecular therapeutics, likelihood of veterinary product development is low, but it is a meaningful approach as we need to be prepared for future emerging unknown viruses. Next, I'd like to talk about possible application of siRNA to other viral infections. As you can see, siRNA is reported to inhibit Hep C and Hep B viral replication, and early clinical study is underway for Hep B. What's noteworthy is the possible application considered for Marburg fever and dengue fever, which is severe endemic infections. I'd like to show one of the studies by Tai et al. On the effect of siRNA on Marburg and Laboon viruses infected monkeys. Together with Ebola virus, Marburg virus is RNA virus that belong to phylofamily. Pathogen is known to cause acute, highly fatal infection of hemorrhagic fever to humans or monkeys. They also designed two types of siRNA against viruses that encode nucleoprotein consumed in MOB and RAV. Each virus has nasally transmitted to rhesus monkeys, and siRNA was intravenously injected 3 to 4 or 5 to 7 days after infection. Survival was observed up to day 28. All animals without nucleic acid died on day 7, whereas the group with siRNA injection showed significant survival improvement despite the fact that the therapeutic injection happened right before the possible death to occur. From these, it is possible that siRNA is effective not only against coronavirus, but also against other highly pathogenic disease. So this is the summary of my talk. siRNAs have potential therapeutics for a variety of viral infection diseases, and they are easily designed and synthesized, and they are powerful measures for emerging pandemic viral disease as a protective platform. In order to achieve the goal successfully, it is important that administration and delivery system be optimized for each disease, and we need to shorten the lead time for GMP and GLP tasks prior to clinical trials. In particular, we need BSL 3 to 4 facilities for in vitro and in vivo studies. Last but not least, I'd like to thank members from Fukuoka Prefecture for collecting various data. Thank you for your kind attention.